Forensic sciences provide the most objective means of discerning the truth when the details of a case are in question. Maintaining the impartiality of the forensic expert witness is especially salient given the complexity of actors in the Philippines' war on drugs. Therefore, both the personal qualifications of the scientist and the context in which they operate must live up to more exacting standards. The role of forensic science services starts at the crime scene with the recognition and recovery of physical evidence. It proceeds with its analysis and the evaluation of the results in a laboratory and the presentation of the findings to the judges, prosecutors, lawyers, and others who are in need of the factual information. From the first respondents to the end users of the information, all personnel involved should have an adequate understanding of the forensic process, the scientific disciplines, and the specialized services provided by forensic laboratories. One of the more difficult challenges faced by the police officers who are working in the field today is accurately identifying the substances that may be illegal drugs. This problem has grown in recent years with the widespread use of ever-evolving novel psychoactive substances such as bath salts and synthetic cannabinoids, which are specifically created not only to mimic the effects of other drugs, but also to avoid being classified as illegal. Assisting law enforcement, scientific advances already play an important role in solving crimes. Laboratories can analyze smaller pieces of evidence than ever before, and law enforcement officials can gain valuable information from evidence that in the past would have been degraded, unusable due to weathering or time. The following methods have been validated by the Scientific Working Group for the analysis of seized drugs. This group consists of subject matter experts who collaborate to determine best practices and develop consensus standards. As such, these methods have been proven to be effective in the analysis of unknown, examination of illicit substances, and are therefore also the best methods to use in identifying unknown substances. Not all these methods are easily accessible in a point-of-care framework, as some require high technical knowledge or a laboratory setting. Therefore, any of the following methods may be suitable on a case-by-case basis. This is due to the fact that some clinics may be able to easily access more discriminatory methods through direct funding or industry partnership, whereas some clinics may have to rely on less precise testing methodologies and equipment due to lack of funding or support. Mass spectrometry measures the precise molecular mass of ions as determined by their mass-to-charge ratio and is the current gold standard in forensic drug analysis. In general, mass spectrometry requires separation, ionization, and finally detection. Virtually any substance can be identified using mass spectrometry in combination with a separation technique. Sensitivity of current mass spectrometers allows for detection of analytes at concentration in the attomplar range. It demonstrates excellent specificity due to characteristic fragmentation patterns, high resolution, and unique filtering abilities available especially in tandem or higher order mass spectrometry. Mass spectrometry produces information about molecular mass and isotopic abundance of elements and temporarily resolve chemical data, allowing for highly accurate identification. Next is the ion mobility spectrometry, which separates and identifies ions based on their speed through a carrier gas. Ion mobility spectrometry is dependent on three molecular characteristics: the charge, reduced mass, and the collision cross-section of the ion. This requires ionization before samples are passed into the instrument. This can be accomplished by ESI, MALDI, APPI, and coronal discharge, or by using radioactive sources such as nickel. A field asymmetric ion mobility spectrometer uses a high electric field to control the movement of the ions through a physical filter. A pulsing electric field can then be applied to select for ions with specific ion mobility. Only ions with the specifically selected mobility will be able to maintain a stable trajectory through the filter. The others will crash into the sidewalls and not reach the detector. So what are the substances which can be detected through ion mobility spectrometer? These are any molecules or any small molecule of illicit substance can be detected very quickly and accurately. The FAME's sensitivity is based on the multiple characteristics of both the ion of interest and the physical environment. So the ion mobility spectrometer can detect one molecule in a billion and is very selective. Its selectivity can be further enhanced when using FAMES, since FAMES is able to operate in environments with high levels of interference with minimal adjustments to operating conditions. So the IMS is a non-destructive and only requires a very small sample if a quantitative method calls for destructive testing. Determination is very quick and can be accomplished in a few seconds, even for a complex sample. Next is the infrared spectroscopy. This is based on the measurement of the amount of infrared radiation which is emitted or absorbed by a sample as a function of wavelength. A spectrum is obtained by passing infrared radiation through a sample and determining the amount of the incident radiation that is absorbed at each infrared frequency. An advantage of the infrared spectroscopy techniques is that virtually all compounds have infrared active vibrational modes and can therefore be investigated both qualitatively and quantitatively. When reference spectra are available, most compounds can be unmistakably identified based on their infrared spectra. According to the SWG-DRUG, infrared can produce structural information that will provide sufficient selectivity that generates the highest discrimination capability. One of the most notable benefits of the infrared spectroscopy is that it does not destroy the sample provided, an important consideration when working with drugs and the people who use them. As well, it requires only a very small sample size in the range of milligrams or less. Additionally, samples can be studied in virtually any physical state. Interference is very common and causes difficulty in identification. Next is the Raman Spectroscopy. It is an optical technique based on the inelastic scattering of radiation after it interacts with matter. The interaction of incident radiation with the molecules of the substance give a spectral vibrational information. The technique involves shining a laser on a sample and detecting the scattered light. A small amount of the scattered light is shifted in energy from the laser frequency due to electromagnetic and molecular interactions in the sample. Plotting the intensity of the shifted light versus frequency gives a Raman spectrum of the sample. An exciting breakthrough in this technology is the development of the handheld portable Raman spectrometers. These devices also search databases in a real time at a device level and give a clear readout of what substance were detected. Virtually any drug can be identified with Raman spectroscopy. It can be used to determine active pharmaceutical ingredients as well as molecules with the same chemical formula but different molecular arrangement and polymorphs. This is important as many of the novel psychoactive substances that have been emerging are isomers, derivatives, and analog of many of the classical drugs of abuse. Being able to differentiate between small differences in physical or chemical structure aids greatly an ambiguous identification. The portable Raman spectroscopy has even been reported to be able to detect the date-rape drug rohypnol in spiked beverages. Raman spectroscopy is rapid and non-destructive. It does not require chemical reagents and can detect separate substances in mixtures and is not subject to interference from water or moisture and, importantly, can detect substance through transparent packaging. Raman spectroscopy is ideal for both organic and inorganic species and can be used for both qualitative and quantitative analysis. Another is the Xray Diffractometry, where the drug sample is bombarded with high-energy X-ray radiation and crystalline atoms in the substance cause incident X-ray beams to diffract in virus detection. This allows for the determination of the spatial structure of molecules by measurement of how X-ray radiation is scattered by the molecular crystal lattice structure. By measuring the angles, intensities of the diffracted X-rays, it is possible to produce a three-dimensional picture of the density of electrons in the crystal, and from this, it is possible to determine the positions of the atoms in the crystals, as well as their chemical bonds and other structural information. So what are the substances that can be detected using this? Any crystalline or partially crystalline substance, such as substances that are solid and usually either evidently crystalline or powder or pill, such as methamphetamine, ketamine, and cocaine, including those in mixtures and compounds with currently unidentified structure, can be identified. This technology is used to identify precise chemical forms but not to quantify them. It can be used to identify diluents or adulterants. This method is sensitive to both polymorphs and contaminants, which is common in illicit drugs. X-ray diffractometry determines structural information of the substance, so the substance can be identified with a very high degree of accuracy. This method is specific because substances have unique diffraction lines or an X-ray fingerprint. It is also sensitive in that drug concentrations and any additional agents used in cutting can be discerned through the obtained data. Studies have shown that this method can be used to identify a specific drug at only 5% of the total pharmaceutical formulation. Another is the MicroCrystalline tests. These chemical tests result in the formation of unique microcrystals of a given analyte when a specific reagent is applied. The unique crystal formation is compared to a reference standard or control using a common light microscope. Microcrystals are compared then based on shape, size, color, and spatial arrangement. So what are the substances which can be detected using this? Several commonly abused substances can be identified, including those of cocaine, heroin, methadone, GHB, ketamine, phencyclidine, methamphetamine, and amphetamine, with test reagents chosen to induce development of specific microcrystals with the analyte and a reference or control standard available. These tests can be highly specific as the crystals formed are a direct consequence of choice of reagent and analyte and are unique under these circumstances. In these cases, a microcrystalline test can be considered highly characteristic but non-specific enough for a confirmatory test. Thus, this method is best suited to pure and separated samples. Sensitivity of this technology is high as samples only require micrograms of substance. The benefit of the microcrystalline tests is this relatively low cost. Minute amounts of reagents are only required. Instrumentation is simple, however, this method does not quantify how much of a substance is present. Unfortunately, the sample that is tested is destroyed in the process, which may be less than ideal for people who are bringing the samples for identification. Another is the thin layer chromatography. This is a technique in which a sample is placed onto a planar stationary phase, then a liquid mobile phase resulting in capillary action. The analyte is either absorbed to the stationary phase or is in the mobile phase, and the time spent on the stationary phase or time spent in the mobile phase determines its retention time. Thin layer chromatography can detect barbiturates, benzodiazepines, GHB, heroin, morphine, opium, oxycodone, and other opiates, amphetamines, cocaine, methamphetamines, MDMA, ketamines, LSD, marijuana, mescaline, synthetic cannabinoids, and cathinone, commonly referred to as bath salts. Thin layer chromatography is a relatively low-cost way to test substances and demonstrates good sensitivity and speed of separation. It can be used as a presumptive test with a fairly high degree of accuracy depending on sample purity. While TLC can identify some known substance in provided samples, it does not indicate how much of a substance is present in the sample. TLC is best used in conjunction with a more discriminating technique such as the Raman Spectroscopy, MS or the IR. Another is the spot or color tests. These offer presumptive testing based on chemical reactions between analytes and indicators. The indicator chemically reacts with the analyte and causes a reaction that creates a certain color staining depending on the analyte tested. Spots are then compared visually with reference charts, the current standard being the Munsell color charts. There is a method that bypasses the human eye and its subjectivity by using a sample smartphone app to identify colors with high precisions and accompanying software that matches the results in a searchable database. This allows for a more precise quantification of the color and therefore higher accuracy of identification. Colorimetric tests exist for most drugs of abuse, including cocaine, various pharmaceutical opioids, amphetamines, LSD, cathinones, heroin, and fentanyl. Actual color results may vary depending on the concentration, whether the drug is in salt or free base form, additional diluents, or contaminants. Positive result may indicate a specific drug or class of drugs present, but not always specific for a single drug or class. Colorimetric tests rely on simple chemical reactions and produce visible results that can be interpreted with the naked eye. Another is the immunoassay. Immunoassay involves the binding of an antibody that is selective for the drug or drug group of interest and a label that will part of the antibody-antigen complex that can be detected using some means such as fluorescence. Antigen-antibody binding is based on a typical immune system response in which antibodies in biological tissue bind to antigens in order to neutralize or remove them. Various opioids and cocaines can be detected rapidly and somewhat effectively using immunoassay technology. Sensitivity is quite high with detection in the microgram range as antibody-antigen interactions occur on a molecular level. Immunoassay is most often employed to detect drug usage after the fact, such as in urine drug screens. Another is the ultraviolet spectroscopy. This method is based on the absorption of light energy in the ultraviolet wavelength range. Light in this range can raise the energy levels of the electrons within a molecule from ground state to higher energy level. Ultraviolet absorption spectrum can be obtained based on the electronic structure of the whole molecule, as this structure will determine what wavelengths are absorbed versus which passes through a sample. Spectrophotometers measure the intensity of light passing through a sample and compare it to the intensity of light before it passes through the sample and capture this information to create a characteristic spectrum. Ultraviolet visible has been used to identify MDMA, ketamine hydrochloride, cocaine hydrochloride, diazepam, phenobarbital, and barbital concentration in the microgram range, as well as specifically identify 6 different compounds and for the first time, accurately discriminate some mixtures. Ultraviolet can be used quantitatively and qualitatively and yields rough structural information, providing modest selectivity to allow for some discriminating capability. So those are the forensic tools used in the investigation of illegal drug cases. Drug testing can be conducted on a variety of bodily substances such as blood, urine, sweat, saliva, or hair, using a variety of tests. So after an initial screening test indicates the presence of a banned substance in a person's body, a follow-up test may be done to verify or disprove the original finding. This second test is known as a drug confirmation test. According to the Substance Abuse and Mental Health Services Administration, for every initial screening and confirmatory test, a chain of custody must be established and followed in obtaining the specimen. Only authorized persons should handle and transport all specimens in accord with the chain of custody, and care must be taken to ensure that samples are not diluted. If the result of an initial screening is negative, no further testing needs to be done. If any specimen yields positive results on an initial screening, however, a confirmatory test of that specimen must be conducted. According to the SAMHSA guidelines, the new test conducted on the initial sample must be completely independent of the initial test and must use a technique that is completely different from that used in the original test. All confirmation tests must be of equal or greater sensitivity than the original test. They should be conducted using gas chromatography or mass spectrometry. The standard cutoff values for the confirmatory test levels have been established on these tests for marijuana, opiates, cocaine, morphine, codeine, phencyclidine, amphetamines, and methamphetamines. Any sample that has a test level above the standard cutoff value is reported as positive. If a drug confirmation test result is negative, however, in spite of a positive initial screening, no further testing is done, and the test is reported as a negative. If the drug confirmation test is positive for a specific substance, then that is considered a confirmed positive result for that specific drug, and again, no further testing is done. Following the collection of samples, all drug testing is done in a laboratory setting. All samples brought into the laboratory are subjected to initial testing, known as the screening tests. This testing is normally done by immunoassay, a method that is less sensitive and less expensive than the methods used in the confirmation tests. Because the method used in the screening tests is not highly sensitive, it is not unusual for these tests to produce false positive results. The method used in the confirmation tests, mass spectrometry and gas chromatography, produce results that are much more accurate than immunoassay. So samples that produced false positives in a screening typically have negative results on this confirmation test. Reasons for false positives on drug screenings may include laboratory errors, antibiotic use, the use of prescription drugs such as ibuprofen or nasal decongestants, and the ingestion of poppy seeds. In cases involving dangerous drugs, the corpus delicti is the presentation of the dangerous drugs itself. Under Section 21 of Republic Act 9165, all seized or confiscated or surrendered dangerous drugs, plant sources, controlled precursors, and essential chemicals must be turned over to the PDEA Laboratory Service for qualitative and quantitative analysis within 24 hours from confiscation, seizure, or surrender. The PDEA Laboratory Service, within 24 hours upon receipt of the items, must issue a certification of its examination results under oath by the forensic laboratory examiner. In cases that the volume of the dangerous drugs, plant sources, or the controlled precursors does not allow the testing to be completed within 24 hours, a partial laboratory examination report may be provisionally issued stating the quantities of drugs still to be examined, but a final certification on the completed forensic laboratory examination must be issued within the next 24 hours. The current jurisprudence on chain of custody in drug cases, as promulgated by the Philippine Supreme Court, involves 4 stages. First is the seizure marking, if practicable, of the illegal drug recovered from the accused by the apprehending officer. Second, the turnover of the illegal drug seized by the apprehending officer to the investigating officer. Third, the turnover by the investigating officer of the illegal drug to the forensic chemist for laboratory examination. And lastly, the turnover and submission of the marked illegal drug seized by the forensic chemist to the court. The third link in the chain of custody is to make sure that the drugs seized, recovered, or surrendered are the same drugs that will be subjected to quantitative and qualitative analysis by the forensic chemist. The forensic chemist must examine the suspected substance, determine its chemical composition, and find out whether it contains any illegal drugs. Likewise, the forensic chemist must also establish the amount of the dangerous drugs present in the substances as well as its purity. The fourth link refers to the turnover of the marked illicit drugs by the forensic chemist to the court when presented as evidence in the case. The Supreme Court in People v. Paharin and Palaya recognized the important role of forensic chemists in the prosecution of cases involving prohibited drugs. As a rule, The police chemist who examines a seized substances should ordinarily testify that he received the seized article as marked, properly sealed and intact, that he resealed it after examination of the content, and that he placed his own marking on the same to ensure that it could not be tampered pending trial. In case the parties stipulate to dispense with the attendance of the police chemist, they should stipulate that the latter would have testified that he took the precautionary steps mentioned. However, the value of even the most carefully recovered and preserved evidence can be lost if the chain of custody is not properly maintained. Chain of custody is often recognized as the weak link in criminal investigation. It refers to the chronological and careful documentation of evidence to establish its connection to an alleged crime. From the beginning to the end of the forensic process, it is crucial to be able to demonstrate every single step undertaken to ensure traceability and continuity of the evidence from the crime scene to the courtroom. Failure to comply with existing laws, rules, and regulations can result in a situation where the evidence cannot be used in court. It is therefore of importance for personnel working at the scene to be aware of and ensure proper compliance with these rules. The equipment required for crime scene work is typically available to the personnel working at the scene, readily put together in a box or kit, and replenished regularly to enable rapid response. Why is it important? Arriving unprepared at the scene, especially without the commensurate equipment and expertise, may result in missed opportunities and compromise the entire investigation. So tackled are the forensic tools used in the investigation of cases involving drugs, how drug evidence are identified, examined, and analyzed, and the importance of forensic science in the investigation of cases.